All right, everyone. Thank you so much for joining us this afternoon for our third and final session of the Science and Faith Symposium. Like I said, we're very excited to have you here with us today and just to explore this really important topic with you that we've been doing the last few weeks. So thank you very much for giving us the honor of exploring this subject with you. If you've been able to attend our past few sessions, I think you'll see it's pretty clear that this is a very, very broad topic. There's bound to be disagreement, as we talked about at the beginning, even among those who share similar worldviews. So whether you're a religious person or an atheist or an agnostic, thank you so much for discussing these things with charity. I think that's the only way forward for us to progress. So in that vein, we'd like to remind you that the purpose of the chat box is to ask questions directed toward the speakers. We definitely want to hear your perspectives, of course, but we do ask that we try to keep the back and forth comments among participants to a minimum, just because that can be a little bit distracting to folks who are trying to listen to the presentations. And we'll definitely have time for interaction at the end with the question and answer sessions. If you do want to have some back and forth discourse about various topics, you can use the Discord server link that we're about to post in the chat. So if you want to chat more with the participants about different subjects, you can definitely use that avenue to do that. So yeah, let us know if you have any questions about that. But thank you so much for everyone for being respectful to one another. And at the end of the symposium, we will be sending links for all of our discussions that we've had, all 6 of them, on both YouTube and podcast format as well. So please feel free to share those with your family and your friends. We'll also be emailing out a resources page if you'd like to explore any of these topics in greater depth and want to dive deeper. And then finally, at the end of our 2 sessions today, we will post a link to a Google form where you can order a t-shirt if you like, and also where you can record your attendance if you need to. We'll also announce the prize winners from our prize drawing. So at the end of each discussion, we'll be giving away a free book authored by one of the speakers and also the $20 Giftogram gift card. So definitely hang on and hope you've got good chances for that. So to dive into our first session this afternoon, has science explained the origin of life? In the 1950s, Stanley Miller and Harold Urey conducted a now famous experiment that some claim to have explained life's origins here on planet Earth. In fact, in his book, Life Ascending, The 10 Great Inventions of Evolution, Nick Lane writes, Miller electrified a simple mixture of gases and the basic building blocks of life all congealed out of the mix. It was as if they were waiting to be bidden into existence. Suddenly, the origin of life looked easy. Some scientists, however, have not been satisfied. Origin of life research and debate surrounding them continues to this day. From a scientific perspective, what do we actually know about the origins of life, and what still remains a mystery? Well, exploring this topic with us today is Dr. James Torre. He is a synthetic organic chemist receiving his PhD in synthetic organic and organometallic chemistry from Purdue University and conducting his postdoctoral training at the University of Wisconsin and Stanford University. He has over 715 research publications and over 140 patent families. Dr. Torre was named among the 50 most influential scientists in the world today by thebestschools.org in 2019 and listed in the world's most influential scientific minds by Thomson Reuters ScienceWatch.com in 2014. He has discussed the origin of life research from a chemistry perspective on several platforms, and we are quite excited to hear what he has to share with us today. So welcome to the symposium, Dr. James Torre.
Thank you so much. I'll just put my slides up here. Okay, so did science explain the origin of life? And so here's my website if you need to know more about me. And I have a fairly new YouTube channel, and I'll be making reference to some of the things that are going on right now on the YouTube channel, because on this YouTube channel, I'm in the middle, I think I just did part 8 of a 13-part series of videos on abiogenesis. And this was in response to a video that was made by a gentleman named David Farina, where he contested a lot of things that I had said about origin of life. So that's in response to him. So they're coming out like every other day right now. And so lots of videos coming out. So there's a big series. So whatever I say here, there's a lot more detail in those videos. But let me just say, has science explained the origin of life? And I say not yet. So not yet can be no, but I like to say not yet. But it might explain it someday. It might be able to do it. And if it does, that doesn't lessen God. I think we would just view God as all the more magnanimous to think, wow, that's how you did it. Um, and that day, though, is far from today. The reason it's far from today is because the more we learn about a cell, the more distant the target becomes, because it's not just a bunch of protoplasm like was thought 100 years ago. It is very sophisticated. And we'll touch on some of that today. We're getting further away in general, while some parts are getting closer to being solved. There are some parts that are getting closer. And for example, uh, the whole issue of homochirality. We're getting more and more knowledge on autocatalysis and how that could bring solutions. Okay, so the videos that are going up on the internet that are coming out, here's the series of videos that are coming out. And I think I just did nucleotides last night, which means not tonight, but probably tomorrow night, there'll be an intermediate summary and it'll go on. So you could just go to Dr. James Torre on YouTube and you would be able to see that. All right, abiogenesis. Abiogenesis is the origin of life from non-living matter. It's the origin of life from non-living matter. And it's to construct any convincing theory of abiogenesis, we must take into account the conditions of the Earth about 4 billion years ago. So whether you believe in a young Earth or an old Earth, that's not the relevant question here, because abiogenesis is defined as the origin of life from non-living matter, and the people who are trying to figure this out are certainly not generally young Earth creationists. So this is the avenue at which I'm coming without trying to make a claim or start a fight with anybody. For synthesis to be categorized as being prebiotically relevant, it must use chemical reagents that are presumed to be available upon an early Earth or accessible to an early Earth. Abiogenesis takes place before biology, before biological evolution can begin. Hence, it is prebiotic or pre-biology. What are the characteristics of life? Because you may remember on the last slide, abiogenesis is we have to make life. Well, here are the characteristics of life, not my definition, but very similar to many scientists' definitions, and it is responsiveness to the environment, growth and change, ability to reproduce, have a metabolism and breathe, maintain homeostasis, and being made of cells. I want you to remember that, being made of cells and passing traits on to offspring. Homeostasis is a steady internal physical and chemical condition. So it's being made of cells. Now, what people are trying to do now is to redefine what life is so that they can say that they made it, going to things as simple as an autocatalytic reaction. But no, life has to be made of cells. This is not my definition. This is the traditional definition of the characteristics of life. What are the 4 classes of macromolecules, large molecules needed for life? And what are the building blocks of those macromolecules? Today, they get all of these from cells. So if you think about both the building blocks and the building blocks of the building blocks, today we get them from cells. So if I say we get those from nature, I mean we get those from cells. But you have to have polysaccharides or carbohydrates. Their building blocks are monomeric sugars, and these certainly encode information. Proteins, their building blocks are amino acids. Enzymes are generally catalysts for synthesis of biological molecules. They're large proteins. These encode information. Nucleic acids like DNA and RNA, and these are a trimer of a nitrogenous base linked to a pentose, a 5-carbon sugar that's linked to a phosphate. In the trimeric form, it's called a nucleotide. These encode information. And lipids, their building blocks are fatty acids, glycerol, phosphate, and often ethanolamine. And what many people don't know, and people are just learning, is that their fluxional assemblies encode information, and these modify throughout the life of a cell. And so you have domains of lipids that move around depending on what the cell is doing at that point. And all of those are information bearing. Now, if you wanted to build this car, if you wanted to build a car, you have all of these different parts. And so this car is partially assembled. I mean, this tire is on the wheel. It has the, the air stem on there. And some of these parts are already assembled. But how many people could, with directions, assemble this? I'm sure there's many mechanics that could. How many people without directions could assemble this? Well, it helps that these parts are kind of laid out the way they are, where you have these seals for the pistons right next to the block here. But what if the parts were all mixed up? Wouldn't it be a little bit harder? And if you had no directions? And what if all of these parts, rather than being in the same room, were spread out throughout the entire Earth? Some on top of the Himalaya mountains, some at the bottom of the sea, some at the North Pole, some at the South Pole. They're all spread out. And you got to find these parts and assemble it. It'd be harder. And when people argue, well, a lot of this came from space, okay, I'll give you that. Now they're spread out all over here. And you got to get all these parts and get it together. This is hard to do. It's hard to fathom doing this. This is much less parts than what you're going to need for a cell. The origin of life. Molecules don't care about life. Organisms care about life. Chemistry, on the contrary, is utterly indifferent to life. Without a biologically derived entity acting upon them, molecules have never been shown to evolve toward life. Never. Molecules don't move toward life. I mean, you can have people pushing molecules toward a living system. Now, we've never created a living system. We've never synthesized a living system that way. But they never even go in that direction. They don't have any desire to go in that direction. So, primordial soup, what's the layperson's understanding? So you got this primordial soup. I've argued that many people still today embrace a primordial soup model, where they think that there was a pond in which there were some simple molecules, lightning strikes, and that formed more basic molecules. Those came together and formed a cell. Those cells came together and formed then even larger organisms that came crawling out of the pond. That's the primordial soup model. Well, and some people say, no, nobody ever thinks that anymore. Well, let's look at it. So this is a survey, and this is done by a Professor Narcombe at Arkansas Tech University. Do you believe the following is true or false? Scientists researching the origin of life have created complex life forms from scratch. That is, in laboratory experiments that approximate Earth's early atmosphere, scientists have mixed chemicals believed to exist before the first life forms to successfully create complex life forms such as frogs. So they're asking people, do you believe scientists have made things like frogs just by reproducing things on early Earth? And out of 715 people, 41% said true. 41% of the people said true. 58% said false. Okay, then they asked another question. Scientists, have they created simple life forms, such as bacteria? For this, 73% say true, 27% say false. Now, who on earth are these people? Well, the average age is 38, so they're in their right mind. The sex is about half male, half female. And interestingly, the highest level of education is 80% of them hold college degrees. So 80% of the people in the West would say that they're embracing this primordial soup model. And for those of you who don't know, humans have never gotten near making a frog or even a simple one-cell bacterium. Not even close. All right, is there hype from the origin of life researchers themselves, or is it just hype from the press? Well, here's something that was written up in the Nature Innovations In section by Jack Szostak, who's a Nobel Prize winner. He's at Harvard University, and he said you can take simple sugars with cyanide derivatives and phosphate, and if you and what the picture he showed here is you put on this heat and light, and what happens is in the presence of UV light and phosphate, RNA nucleotides were formed. That is totally untrue. He's making that is totally incorrect. He's making reference to work done by John Sutherland in which there were, I don't know, 15 steps to do this sort of thing. And it wasn't from iron cyanide. These things have very little free cyanide in them. This is 1,2,3-propane triol, and this is ethylene glycol, which he called simple sugars, but that's a minor error. You'd have to put an aldehyde here for them to be considered simple sugars, but nonetheless, and this is not really ribose, because ribose, you have to show stereochemistry or else it's not ribose. But in any case, this is what's put out there. This was published in Nature, which is the top journal in all of science. And this is written for the scientifically literate, not working in the origin of life, for the scientifically literate. And this is a huge extrapolation to say such a thing. There's so many steps in here that are hard to ever presume would be prebiotically relevant to get something like this. But nonetheless, it's shown in a single step, and it's described in this way. So that is a lot of hype. Here's even higher level extrapolation by a great chemist, John Sutherland, whom I really admire as a synthetic chemist. This was written up in Nature Chemistry. He wrote that all the cellular subsystems could have arisen simultaneously through common chemistry has raised the, and I'm saying that he wrote all cellular subsystems could have arisen simultaneously through a common chemistry. And what I say is that has raised the level of supposition from mere molecule types to now cellular subsystems, where molecules are working in concert toward a common functional goal. Now, I work in the area of nanotechnology where we try to get molecular systems working together in concert, carrying out a function. It is very, very hard to do that. And he showed a very simple set of reactions, and he made the claim that now all the cellular subsystems could have arisen simultaneously. I don't know how he got the simultaneously out of his presentation in that paper through a common chemistry. And a cellular subsystem, I mean, this means many, many molecules working together in concert toward a common goal. So this is the kind of hype that has given the layperson the presumption that a prebiotic pool could have made all these things, and you just can reproduce that prebiotic pool in a lab and start making frogs, or if not frogs, certainly bacteria. It's this kind of hype that has influenced the field. So my call to my colleagues like John Sutherland, I would say that we've got to tone these down, because the press takes this and amplifies it all the more. But this is the scientist statement. This is surely something that we can control. This was written up in Science. Science said, biologists create the most lifelike artificial cells yet. And then when you read the paper, they're referring to an article that came out by Devaraj in Nature Communications, where they took plastic beads made in a fabrication facility. So you do soft fabrication, you make these little plastic acrylic beads, and you take these beads and you fill them with clay. So you actually make the bead around the clay. Clay is positively charged and DNA is negatively charged. So it will go in, and then they'll introduce DNA. The DNA will stick to the clay, go in there. And then you add materials just like you buy in a kit. You can buy kits to make proteins. So you can buy the DNA, you can buy the RNA, and you can make proteins this way. And so they essentially took all these things either from a cell or from a kit, they put it in the solution, it diffuses into these semi-porous acrylic cavities, and you start making proteins. And the proteins leach out. And they call this quorum sensing because they said the ones nearer could get proteins that had leached out of other ones of these cavities. We used to call this not quorum sensing, we used to call this diffusion. That as things leak out of one thing, the things nearer to them would get more of the material that had leaked out. That is called normal diffusion. Things that are closer get more of it. It was termed quorum sensing, and these were the most lifelike cells yet. And this is how it was described in the journal Science, one of the top 1 or 2 best journals in the world. And this is how it was described. Now, maybe they're right. This is the most lifelike artificial cell yet, which means that we're very far from making a cell and an artificial cell that is anything like life. And you can just buy a kit to do this chemistry. This is just chemistry. It's not life. It's just chemistry. You can do this in a test tube, do it in labs all the time. This was written up in Quanta magazine. There was an article written by an MIT physicist who's now at Georgia Tech, Jeremy England. And what he is quoted in saying regarding his article, this is what England himself said: you start with a random clump of atoms, and if you shine a light on it for long enough, it should not be so surprising that you get a plant. And that's just not correct. I mean, people have shown lights upon atoms for many, many years, and lights of all different frequencies. I mean, just the whole electromagnetic spectrum. A plant has never formed out of these things. But this is what the researchers themselves say. So imagine what the press does to ramp this thing up, and this is what causes the confusion to the general public. Homochirality is a big issue. Homochirality is where you can get mirror images that are non-superimposable. As long as you have 4 different groups around a carbon atom, that'll give you a stereogenic center, and you'll have homochirality. And where are we on homochirality? Well, enantiomeric resolutions are difficult experiments to execute. There's massive human involvement in the experiment. And even in autocatalysis is where you take a little bit and it becomes a catalyst. And if you look at autocatalysis experiments, the ones that really work well are never done in water. They're done in water-free systems. And there's massive human involvement. The interpretation of data is not trivial for the researchers, and it's not clearly presented often in their own data. The interpretation by the reader is made all the harder by often insufficient data provided by the origin of life researchers. It can be hard for the scientific research articles, especially for the layperson, to understand, and the layperson often doesn't have the expertise to properly interpret the data for herself, and therefore her descriptions or contextualization suffer. So she relies on the press descriptions, which can be inaccurate, as we've seen many, many times in the press. But homochirality, there are these theories on how homochirality could have come about. And generally what you're dealing with is you're dealing with a single stereogenic center in the experiments that are being done. But people are getting closer and closer to having good descriptions through autocatalysis. The autocatalysis reactions are not generally run in water. But I think we'll have solutions to this someday. It's not been solved. We are far from getting it solved in any prebiotically relevant manner. But there are many interesting proposals on the table, and we're far from having a description for this today. But maybe in 50 years, we'll be doing a lot better with this. Making single enantiomers is one of the hardest problems for all synthetic chemists, not just those doing origin of life. All right, you have to be able to have carbohydrates, which are sugars or saccharides, and the polymers therefrom. So this is the video. I just grabbed this from some other slides that I had. This is the video on Professor Dave Explains site, where he came up with this video calling it the Elucidating the Agenda of James Torre Defense of Abiogenesis. So that's why my series just started coming out in February, 6 months after his. And I don't address his agenda. I don't psychoanalyze him. I don't reference his faith. I don't pick on his faith. That's irrelevant to me. I just go after the science in my videos. And so I don't do any ad hominem attacks. If I do, that's by accident, and I'm sorry. I'm not trying to do that. So some think that carbohydrates consist of the same monomer over and over again. That's what was said on this video. And some say any biomolecule is easy to synthesize, which is and it's people like this Professor Dave saying this that make the layperson think, oh, this can be easily done. Now, I have colleagues here in the university that work on natural product synthesis, and in fact, my PhD, a lot of it was in natural product synthesis. To say any biomolecule is easy to synthesize is very, very strange thing. I have colleagues that will spend 50 person years on trying to duplicate a molecule using modern synthetic methods, duplicate a molecule that cells will be banging out quite easily. And there's many molecules that we just can't make. And then to think of having it done in a prebiotic sense is all the more difficult. And so here's some naturally occurring monosaccharides, and here's some representative homopolysaccharides. So there's a lot of different sugars, and they can be hooked up in different ways. You can have the 1,4 linkage, or you can have the 1,2 linkage, or you can have alpha and beta, and all of this is characterized by why does this hydroxyl group not attach here? Why is it this hydroxyl group? Why not this one? And why not this one? This one's on a primary carbon. Each of these is on a secondary. So you would think that this might be more reactive because it's on a primary. Well, this is the glycosidic linkage, and there's reasons for that, but these can be constructed in all sorts of ways. And they're not just homopolymers. You have lots and lots of heteropolymers where you have different carbohydrates put together. These are exceedingly complex. If you want to try to make carbohydrates, it's really hard. For example, Eschenmoser wanted to make ribose because from this he wanted to make DNA in a prebiotically relevant manner, and he was just going for the racemic material. He wasn't even just trying for the homochiral material. Just even the racemic was good enough for him. But of course, you would end up with all of these different diastereomers. And so, because there's 3 stereogenic centers, if it's in the closed form, you're going to have 4 stereogenic centers. And this is very difficult chemistry to do. Making carbohydrates is so hard. And to think of how it was done in a prebiotically relevant manner is really hard. Yes, you can do this with modern synthetic techniques that would never have been available, and using protection, deprotection schemes and solvent systems that would have never been available on an early Earth. But even then, carbohydrate synthesis is hard. In fact, within the realm of synthetic chemistry, carbohydrate synthesis is among the hardest things to think about doing. We'll see why in just a moment. These are the foremost reaction, which is used to make the polyols. You get competition by the Cannizzaro reaction, which decomposes these under the very same reaction conditions in which they form. And my argument is time makes abiogenesis less likely rather than more likely because molecules degrade. So molecule degradation is a big problem. And you can see that all the time. Polymerization, if you're making saccharide, if you make a sugar, making the sugars wasn't hard enough to make the polysaccharide, to hook them together is just as hard. And here's the problem. If you had, for example, nucleotides, if you had all nucleotides with the A base, how many different ways could you hook that together? All the same nucleotide. Well, there's one way, A. That's it in a nucleotide. That's the hexameric nucleotide, oligonucleotide. Well, if you had all the same all the same repeat units of a reducing hexasaccharide like D-glucose shown, this can form how many different combinations of assemblies can this make? Over 1 trillion. Over 1 trillion different ways that this can be hooked up. You go, what are you talking about? It's just the same one together. No, because you can have this hydroxyl site hooked to there, this hydroxyl site hooked to there. Each one of these anomeric centers can be alpha or beta, alpha below, beta above, or any one of these. So it has all of these different tentacles. So how do you hook these together? So why should this 4-position react here rather than the 3-position, or the 5' position, or I guess that's technically the 6th position. Why not this one? Why? And that's the difficulty in carbohydrate synthesis. So if you look at the way chemists build carbohydrates, you have to selectively protect each alcohol. Well, how do you selectively protect each alcohol versus another? Well, you have to protect these two together, then protect these two together, then functionalize that one, then deprotect these two. And then it's difficult, hard chemistry, lots of protection, deprotection. And here's the reference on that. So carbohydrate chemistry is really hard. So when people talk about the Miller-Urey experiment, they will never talk about carbohydrates in this because there's no easy way to think about doing this, because you don't have just one stereogenic center, you have a lot of them. So this is hard chemistry to do. So carbohydrate synthesis is exceedingly hard. Coupling carbohydrates together is painstaking. The untrained have no idea how hard the syntheses are to perform. That's why they think that you can just you get a primordial soup, it all comes together. Trained remain clueless on how these could have formed in a prebiotic system. The untrained see little difficulty because they don't understand the complexity. All right, building blocks of the building blocks. How clueless are scientists regarding the syntheses of the 4 classes of compounds needed for the construction of a cell? Well, remember I showed you that car, and that car had a bunch of parts? What if I told you not to take those parts and make the car? I said, no, I want you to make the car, but you can't have the parts. No, you can't have the parts. You got to make the parts. Yeah, you got to make the parts. And all you have is what's available on Earth. All you have is aluminum ore. You have to take aluminum ore, and you've got to extract from this, you've got to get from this aluminum oxide. After you get the aluminum oxide, you've got to take a carbon electrode, and you have to reduce the aluminum oxide to aluminum metal with a low voltage, high current, and you have a sacrificial carbon electrode which makes CO2. You've got to be able to extract the copper from copper ore to make all the copper wiring. Iron ore, you've got to be able to get the iron out of this and make steel. You've got to be able to take crude oil, which would not have been available on a prebiotic Earth because it's believed that this is of biological origin. But let's just say this were available. How many people know how to take crude oil and make all the plastics that you need? There's about 800 pounds of plastic in a modern vehicle. Now, I'm sorry, 800 kilos, which would be about 450 pounds of plastic. So how are you gonna make all the plastics? How many people know how to convert crude oil into plastics? How many people on Earth know how to really do this? Not many. And then how many people know how to do all these other things? There's probably not 5 people on Earth that know how to do all of this to make the pieces that you need just to start making those parts. That's what early Earth is stuck with. It's not that you can go to the store and buy, that you can order online and buy the carbohydrates and buy the nucleotides. All of those came from cells, which were already there. You got to start with this stuff. This is what you got to start with. So the building blocks of a cell, you got to have the polysaccharides, you have to have the proteins, you have to have the nucleic acids, you have to have the lipids, and all of these already, when we are doing origin of life experiments, these so often come from living systems already, so it's cheating. What if you had to go back right to the beginning and start with the very basics? And because what chemists do is we start with cylinders and all the gases are separated and compressed for us. How convenient. On early Earth, all the gases are mixed together. And then in all the stereo-scrambled syntheses that origin of life researchers do, all their reagents are nicely separated. But that's not what you have on an early Earth. What you have on an early Earth are presumed these sorts of chemicals: ammonia, methane, oxygen, carbon dioxide, hydrogen sulfide, water, sulfate, formaldehyde, carbonate, formate, hydroxide, cyanide, cyanate, metal oxides, and so forth. So this is what you've got, and this is all you have. You're out there, you've got to find all these things and do the synthesis. Look at what early Earth was stuck with. You have to find it all out there, and you only have rocks. How are you going to find all this out there? Look at what early Earth was stuck with. So we have no idea how this was done. Then you have peptides, which are also proteins, and amino acids are the basic building blocks. And this is what's shown to people, that you can just take amino acids and they hook together in water. This doesn't happen. This is a condensation reaction that's forming water. I mean, if you don't have enzymes around where this is coming out in the cleft, that this doesn't just happen in water, it goes back faster than it forms. And they say, you know, it could, it possibly, it could, perhaps. And all of this nonsense comes out there, which isn't real. Those who do chemistry for a living know how hard this is. And you see how conveniently these are all R groups. He's not showing any of the functional amino acids where you have the reactive side chains. And that's why even origin of life researcher Matthew Pauner published in Nature in 2019, he says, peptides are essential for life, and they're orchestrated. The biosynthesis is orchestrated by a complex host of genetically encoded enzymes. It is inconceivable that these sophisticated and coordinated macromolecules suddenly emerged at the origin of life. Peptides are widely assumed to be the products of amino acid polymerization reactions. Whilst conceptually simple, in practice, there are good reasons why these reactions are ineffective in water. So it may be that Miller-Urey is a red herring, that we get all of these amino acids and think that those amino acids start forming in water. Why haven't we ever been able to polymerize those in any prebiotically relevant reaction? Because they don't polymerize, because they're zwitterionic. They exist as a plus and a minus, and in water, they're very slow to polymerize. They go back the other way. So maybe peptides didn't come from amino acid couplings. And that's what his article in this Nature issue is about. He has a different way of thinking about how polypeptides may have formed. There's no control of the stereochemistry, yet it's a very clever approach, very clever approach. And maybe Miller-Urey just threw him off for the last 68 years. Here's what you have to do for once you have the amino acid, and you look at solid phase synthesis, you first have to have a resin with a linker. You buy the amino acid, which came from a cell. You have to have a side chain protecting group. You have to block the side chain, and you have to have a nitrogen protecting group, and you load that onto the resin through the carboxylic acid. Then you have to deprotect that without deprotecting this. And then you have to buy more of your next amino acid that'll have a different side chain protecting group, if it has a different side chain, and another nitrogen protecting group here. Put this on via the carboxylic acid to get the dimer, and then you have to wash all the things that didn't react and do this. So this is hard to do, and this is not done in water. These things don't go on the zwitterionic forms. All of this is hard to do. And then you have the nucleotides, the DNA and the RNA. And so for RNA, you have so here's your nucleoside, here's the base, and here's the sugar. If you have a phosphate there, it'd be the nucleotide. RNA has the two hydroxyls, DNA is the deoxy, the 1-OH deficient. They both have bases, they share some bases, and some are unique. But in any case, again, this is hard chemistry to do throughout. This is how solid phase synthesis is done. And so you have to put it on a bead. They come in DMT protected, and you buy this, you buy it with the base on there. This was all made in a cell. This wasn't made by any prebiotically relevant manner. Then you have to deblock and then bring in a phosphoramidite, all human-made constructs to get this coupling to go. And then you have to cap the ones that didn't grow and uncap. And so these things are hard to do. And so there's 4 basic steps here, 4 basic steps per cycle. And if you want to build 100 mer, I mean, if you're 99.9% efficient, it's going to be hard to build 100 mer cleanly. You can make up to 100 mer, but usually you make smaller ones and hook them together. But in any case, let's say you had RNA. Everybody wants to use RNA, and they want to use RNA and say, oh, we just have RNA and that'll do it for us. How is that? If you just look at RNA, this is right off the Sigma Aldrich website, or Millipore Sigma website, that says that for storage of RNA, you have to have it in a tris ethylene diamine buffer, And you have to store it at minus 80 degrees, which minus 112 Fahrenheit. Why? Because they degrade. Even at 0 degrees, their degradation is very rapid. I'm not talking about enzymatic. I'm just talking about this hydroxyl hitting that phosphate and coming off. And you want to say that these things can duplicate themselves? I mean, how do they duplicate themselves without other enzymes? And RNA has only been known in the most loaded systems to duplicate about 10% of itself. And the daughters were never long enough to further duplicate themselves. So this whole thing that RNA just duplicates itself, I mean, in well-designed and primed systems, they only duplicate about 10% of themselves. And plus, they fall apart quite easily. It's said that these things come together on hot clay. Well, 20 to 80% of the time when it comes together on hot clay, you get not the 5' 3' linkage, but you get the 5' 2' linkage. So 20 to 80% of the time it's wrong. And that throws off all translation and transcription, and those act as siRNA. So again, here's this thing of people saying that time is not a problem, and I've always argued that time is a problem. Here's an example from Sutherland's work, where he was doing what he called prebiotically relevant synthesis, where he was trying to put amino acids onto nucleosides. And so here's his amino acid, but conveniently it's N-acetyl protected. Did you find N-acetyl protected amino acids on early Earth? This is supposed to be prebiotically relevant. Moreover, that stereogenic center got racemized during his reaction. But what I'm trying to point out here is even in his prebiotically relevant synthesis, at most of the pHs, he got 0% yield. At one of the pHs, so at pH 5, he's able to get 4.7% yield, and that's reached after 12 hours. Right away, he has to fish that thing out, because if he doesn't, after an extra 48 hours, that has gone from 4.7% yield to 2% yield. The very reaction conditions in which you make many of these molecules are the conditions at which they decompose. So that's why the chemist goes in and fishes it out. That's why I say time. Just say, well, you had lots of time. You had lots of time. It goes bad under the very same conditions that it was formed. And so you know, some say it's very easy to make these molecules. I argue it's very hard to make these molecules. And just because you have machines, it was said that this is either a protein synthesizer or DNA synthesizer. This is an HPLC, I think. But in any case, just because you can program something into a machine where you've bought all the building blocks of the building blocks, the basic constructs, you've bought them from companies where those came from cells to begin with. Even if you have those, and they're all human-made protected, because you can spend millions of person years, or certainly hundreds of thousands of person years doing nucleotide chemistry and doing protein chemistry and thinking about how to do this, and billions of dollars on equipment design. Because you can do it with equipment doesn't mean an early Earth could do it. They didn't have this equipment. All right, so let me just skip through this. Lipids in the cell membrane. Well, lipids must be easy to make, right? No, they're not. Because what you have to do with lipids, they all have a stereogenic center here. And these are the simple lipids. Sphingolipids have 4 possible stereoisomers. And this is not including whatever cis bonds you might have here. Cis or trans bonds. Just take this as an aliphatic chain. And when you start with glycerol, 1,2,3 propane triol, you have to desymmetrize this. So the 2 hydroxymethyls are enantiotopic. And so you have to be able to pick out one over the other. And so this is a big problem. And how does it desymmetrize this? We don't know. Very hard to do this. Even getting a fatty acid is hard to think about how that would have formed on an early Earth, because you don't have long chain hydrocarbons. You want to use a Fischer-Tropsch synthesis with carbon monoxide? This stuff is hard. You want to use aldol synthesis to make fatty acids? That's really hard. And how are you going to get the thing fully hydrogenated at the end? So before you have biology, it's hard to even think of how you're going to make a fatty acid from methane. I mean, really hard to think about that. And then if you look at these phospholipids, you have the polar ends out here, and the non-polar ends in here. One is inside the cell, one is outside the cell. But these are never symmetric in biological systems. If they are symmetric, you don't get the proton gradients that you need for life. And all protocell experiments deal with symmetric systems, and all living systems deal with non-symmetric. We have no idea how you get this non-symmetric. You can do it when you use what's called flip-base enzymes, but enzymes weren't yet around. So how does it do it? We don't know. We have no idea how that was done. Some people say that this is easy to make. Not when you have a stereogenic center here, and not when you have to desymmetrize this. It's very hard to make. And the improper stereoisomer of lipids are known not to function in cell operation. So it's not that you can just use a racemic mixture of these things. So also this idea that fatty acids alone could have been the cell membrane. Devraj, who's an origin of life researcher, argues with this. He says, quote, all living systems harness proton gradients across phospholipid membranes to generate energy. Vesicles formed from simpler amphiphiles, such as fatty acids, are unable to maintain proton gradients, suggesting that diacyl phospholipid membranes appeared early in the origin of life. So in other words, your fatty acids, simpler amphiphiles like fatty acids, are not able to maintain a proton gradient. They will not do for the lipid bilayers that are needed around cells. And one cannot just suggest a spontaneous synthesis of fatty acids. Spontaneous is the code word that people use when they have no idea. Oh, that spontaneously arose. Oh, okay, I'll accept that. No, what do you mean by spontaneous? Tell me. I mean, some chemistry must have happened. How do you envision that happening? So this word spontaneous is a code word for I have no idea. All right, so summary is that there's no control of absolute stereochemistry and homochirality. There's little control of relative stereochemistry in any of these prebiotic syntheses. There's little to no accounting of yield. How do you say you even had a kilogram of something? Once you take a kilogram through 10 steps, you might only have a milligram. And there's no optimization. There's no accounting of purification. You can't run on just crude mixtures of things. There's no accounting for mass transport through the sequence. There is enormous human intervention in every phase of the experiment for origin of life researchers. A prebiotic Earth would have none of these luxuries. The origin of life researchers, those superb synthetic chemists, are generally silent on these essential synthetic questions. Reading the articles by origin of life researchers leaves enormous holes which render their work prebiotically irrelevant and misleading the non-synthetic chemist. This is not for lipids only, it's for almost all origin of life researchers' syntheses. I mean, there's enormous holes in this where you scratch your head and you say, how is this prebiotically relevant? You can't even use a vacuum pump if you want to match a prebiotic Earth. I mean, it's just hard to do. All right, and then there's chiral induced spin selectivity. Some say chirality came as an afterthought. Life then came, and then things got purified out by enzymes. Well, you know, think of a bacterium. We've not figured out how to assemble a functioning cell. We've not figured out how to make the 4 building blocks. And so here's the 4 building blocks: carbohydrates, peptides, nucleic acids, and lipids. We've not figured out how to make the building blocks of the building blocks. And there they are, the monomeric sugars, the amino acids, the nucleotides, and the glycerol. You may say, well, we can make most of these by milli-Urey. Okay, I'll give you that. That's fine. But we don't know how to get these to polymerize from here to here. We've not figured out how to address the homochirality issue. This was all racemic. Yes, asparagine can self-crystallize with one enantiomer, but the others cannot easily. Yes, there is this this autocatalysis that can help, but to find an autocatalysis that's relevant in water, we have yet to really figure that out. We've not seen that. We don't know how to polymerize these. We have no idea how to polymerize these to make what we want to be able to make. We have no idea how to desymmetrize these. So all of these are stereogenic centers. These are enantiotopic hydroxymethyl groups. And therefore, did homochirality occur after the simple cell formed? Well, did it? Well, we know from chiral induced spin selectivity, which is a phenomenon that's only been, people have only discussed for about 15 or 20 years now, and it's getting more work recently. It's just amazing, just amazing. How come nature is so selective with its reactions? It has like 99.999% yield on its reactions, and we go in a lab, and if we have 90% yield, we think it's great, and that's after a lot of optimization. And we are taught, students are taught, all of this happens in enzyme clefts, and you get this hand in glove sort of match that makes the molecule fit and the other enantiomer doesn't. That's only part of the story. What chiral induced spin selectivity tells us is that chiral molecules, the chirality that's in molecules, acts as a spin valve. So it selects one spin. So if we're thinking of hydroxy radical, lots of hydroxy radical in our bodies that is there. We think of hydroxy radical, period. In biological systems, that electron, in any system, that electron right there is either spin up or spin down. The chiral system selects one of those to allow transport through it. It is a spin valve. And because of that, if you go through a singlet surface in an anti-parallel state, you go through a singlet surface, you get hydrogen peroxide. If you go through the parallel, the triplet surface, you get oxygen. This is how, in a biological system, it can make 100% oxygen or 100% hydrogen peroxide. We have no idea how to do that as chemists. We're just learning how to do that. Because you put chiral systems in there, it influences how that thing goes. Why don't cells overheat? People have looked at this. Cells are very high energy systems, low entropy, high energy systems. Why don't they overheat? For an electron of the proper spin, chiral molecules show far less backscattering of the electron. This in turn greatly reduces the heat release from the molecules during the electron's passage. Lower heat affords any cellular system an advantage. Hence, homochirality is required. You would have too much backscattering, so nature selects only the molecule with the right spin and allows that through, and because of that, you don't get backscattering. So you had to have a chiral system there already. If you just calculate just what happens in the brain, here the calculations have been done. If you had the capacity of a human brain, it would need at least 10 megawatts to operate. This is equivalent to the output of a small hydroelectric power plant. The human brain uses only about 10 watts. Surely some thanks are due to homochirality. So this homochirality cannot have been an afterthought in cells. It had to have been there in the beginning. So anyway, let me go through this. Constructing a cell, you have the whole cell assembly problem. People will talk about miliurie, but they won't talk about getting that sugar in homochiral form. They won't talk about getting these sugars in homochiral form. They won't talk about distinguishing between the homochiral hydroxymethyl groups to make lipids. They won't talk about what's involved in making these polymers. We talked about you get a trillion different possibilities to make a hexamer. These things are hard to polymerize. These things don't polymerize in water. You want to try to get this thing to polymerize? How well does this go in any prebiotically relevant manner? I mean, articles are coming out just in the last few months on this, and they make like tetramers, which is an absolute joke. I think they made a hexamer, which would be 2 codons. You say this forms on hot clay. Well, how hot? How hot is the clay? Because remember, this hydroxyl swings around and hits that phosphate and off it comes. These things have to be stored in a buffer at -80 to keep them from decomposing because of this reaction. And the residue order. So these things have to hook up. You can get codons, but they're not describing for anything relevant to life. There's the interactomes. If you just think of protein-protein non-covalent interaction, we're just talking about the non-covalent interactions between the proteins in a single yeast cell. Just the non-covalent interactions between just the proteins in a simple yeast cell. The number of arrangements you can have is 10 to the 79 billion. Yeah, 10 to the 79 billion. Here's, that right, chopped right out of the paper because people say you must have had that number wrong. There it is. Now mind you, there's 10 to the 90th elemental particles in the universe. That's a big number right there. These non-covalent interactions of how molecules are paired up and lined up, this is what allows what's called the electrostatic potentials to go through for information transfer. Electrostatic potentials is where you modify one, you bring up an electric charge here, it modifies this orbital, which modifies the next and the next. These are non-covalent interactions between interacting orbitals. These travel at near the speed of light. They're using extremely low amounts of heat release on this. And this is why a cell can survive. But you have to have this ordering, and that's why you can't easily dehydrate a cell and take all the structural water out of that and then rehydrate it and ever get it to work. Once you've removed the structural water, it's gone. And that's why cells pass on information by putting it on each half and breaking on down like this. This is amazing. There's all this order in cells. And then people will say, well, the cells used to be simpler than they are now. Well, how simple were they? You can do the calculation, because right now, the smallest genome known now to grow, capable of autonomous growth in a laboratory is 580 kilobases, 580,000 bases. Well, there's been calculations, and the lowest you can get, the theoretical limit is 206 genes, where a gene is a distinct sequence of nucleotides forming a part of a chromosome, the order of which determines the order of monomers in a polypeptide or nucleic acid. This is what you have to have in a minimal cell. DNA replication. This is not me. This is biophysicists did this calculation. You have to have DNA replication, repair, restriction, and modification, a basic transcription machinery, amino acyl transfer RNA, transfer RNA maturation and modification, ribosomal proteins, ribosomal function, maturation, modification, You got to have all of that. So when people give you this garbage that with cells were much simpler, and then you can cite all you want that the earliest cells we see are not much different than the simplest cells we have today, they don't believe you. Well, here's the calculation. It has to have all of this, and this doesn't include that with this little bit of set, you still have to have all the 20 amino acids coming in from some other source. And you say, well, it came from outer space, that this gave you all your building blocks. Fine. Even if you have that, only trace amounts are there. And they generally have only small amounts of enantioselection. There are some that have claimed higher enantioselection in this one, which is not even biologically relevant to human beings or to any mammals on Earth. But in any case, this is contested. But even if I gave you that, I'll give you all the chirality you want. All the chirality. I'll give you, and here's the challenge. A challenge awaits. You will get a Nobel Prize if anybody could make a working cell, not a complex cell, even just a simple minimal cell that shows the functions of life, all right? If you could do this. So what you have to do is you have to take the simplest cell and you have to make all of these things and make the simplest cell. And you go, no, that's too hard. Okay, what I'll do, here's the challenge. I'll give you these with all the enantiomeric centers already set. I'll give you these. I'll take these from a living cell and give it to you. Could you polymerize these? Please, and make me what you're going to, no, this is too hard to do. Okay, I'll give you these, and I'll give you these in the order you want. You tell me the order you want, you got it. I'll give you all of these. In fact, I'll take them out of a living cell. You tell me what cell you want, I'll take it all. I'll give it all to you. Can you take these and construct a cell? I'll give you all of these pieces which you already got. Can you construct a cell? I'll give you all of the parts to that car. Here they are. Do the construction. Well, you can't do it? Come on, a Nobel Prize awaits you. What's wrong? Even a mindless early Earth was able to do this. And it had to make all of these starting materials from methane and formaldehyde and things like that. And you can't do it in your lab. You can't be better than a mindless Earth. That summarizes the state of assembling a cell. So if some people say, well, we know how the chirality, okay, I'll give you that chirality. I'll give that to you. So when you see articles like this, researchers solve the puzzle of origin of life on Earth. Wow, what a claim. I have to look that up. And you look it up, and here's what they've done. They've taken cell membranes, and they've bought all these components which were taken out of cells, taken out of living cells, and they've mixed it, and they've gotten all this to go into this membrane. And that's what they called in this article, researchers solve the puzzle of the origin of life. It's not that. What they've done is they've taken all these parts and they've shoved it into that car body and they say, hey, I made a car. You didn't make a car. You took parts that came out of another car and you shoved it in that car. And it still doesn't work because they're not functioning. You have to have all that interactome, all that covalent and tons of non-covalent alignment. So in summary, we remain so far away from figuring out how to build even the simplest of cells, and the targets move further from us every year because the complexity of the cell, we learn more about it. Contrary to the claims of the naive, every step is mind-numbingly hard. It is so much more than just making a strand of RNA. Biologists are sadly confused on that point. Realize that people are wrong when they suggest that we're close to solving the construction of even the simplest cell that can be conceived, wherein it meets the minimal requirements of a cell as we know it, which I've given to you. Humankind remains clueless on the origin of life, but that does not mean that we will forever be clueless. I could never say that as a scientist. If you're a graduate student, think about these things. And to origin of life researchers, you see how lost people are, even the scientifically literate, like that YouTuber on the true state of science. Help me out here, please, on getting the record corrected by passivating your unnecessary claims in your articles and to the press. I mean you no ill will either. You are my colleagues. So seems to have frozen up here. There we are. So anyway, here's problems that I think are worth studying in origin of life. The origin of the informational code, mass transfer problem, the origin of homochirality, Leventhal paradox of protein folding. A lot of studies going into this, but also Leventhal paradox 2, which is for interactomes, a method to remove human intervention, the secret to syntheses that can be repeatedly survive impurities or solve the separations problem, and the undirected synthesis challenge. So, postscript. I've written on origin of life. I have 5 articles on origin of life, and let me see if I've got here. So I've got 5 articles. If you just Google James Torre inference, you'd find some of those. Those that love God need not feel threatened by origin of life research. And the reason I say that is, even if we figure this thing out, if scientists figure out the origin of life, it doesn't lessen God. We'll just say, oh, that's how God does it. He's all the more magnanimous in our eyes. Just like when we figured out DNA structure and how the genetic code, that doesn't lessen God. We go, wow, God, you're all the more amazing. That's amazing. When we do learn how life might have originated from a scientific perspective, it doesn't bother me. It doesn't bother me at all. We cannot expect the materialists to readily embrace what we hold so dear. I mean, there's things we've got to do. And that's why I tell you right now, if there's anybody here who is not a believer in Jesus Christ and his resurrection, you send me an email to tour@rice.edu and I will gladly share why I believe that Jesus Christ has risen from the dead. You just send me an email and I will be glad to share with you. One-on-one, we'll set up a Zoom conversation. Has science explained origin of life? Not yet, but it might explain it someday. That day is far from today. We are getting further away in general, while some parts are getting closer to being solved, but further away in general because the cell is getting more complex, because the goalposts are moving further away as the cell gets more complex. And that's where I end, and I'll open it up for questions.
All right. Dr. Torre, thank you so much for that mind-opening talk. I know some of it went over my head, but I still learned a lot, so thank you for that. So the first question is, what is a good description of the information that is stored in the 4 prerequisite elements of a cell? For example, could you please elaborate on how lipids carry information?
Yeah. So there is work by Tony Futterman at the Weizmann Institute in Israel. And what you have is there are no cells with just one lipid type. The vast majority of cells have many lipids. Over 40,000 lipids have been identified in cells. I mean, so there's a lot of different types, but any one cell will certainly have hundreds, if not thousands, of different lipid types. And the lipids form domains. So they form domains. So you'll have a domain of similar lipids, and then another domain. And so these are like tiles. And these swarm where you have like tiles starting to come together. They'll form swarms, and then they'll move across the surface of that membrane, depending on the function of the cell, what the cell is going through, because so much electron transfer occurs in cells. So these are dynamic systems, and there's information of what's happening in the life of that cell embedded in the domain swarming of a lipid. That's how I mean information is stored. That's non-covalent storage of information in a lipid. Did that answer the question, or was there another part to that I missed?
No, I think that answered it.
Okay.
So the next question is, Dr. Torre, could you define life? Is there a scientific consensus on how to define it?
Yeah. I put that in that was like in my first slide. I mean, this is a definition that's not just my definition. This is a definition that's well defined, because nobody's going to believe me. I mean, why should you believe me? I mean, can I be believed? I mean, you don't have to believe me. Here, what are the characteristics of life? Now, I don't know if this is answering your philosophical question of what is life. I don't know. All right, let the philosophers at Ratio Christi do this. That's what they specialize in. But this is the scientific definition: responsiveness to the environment, growth and change, ability to reproduce, have a metabolism and breathe, maintain homeostasis, and being made of cells and passing traits on to offspring. Some people include in this, it has to be able to evolve. Okay, so that's in some definitions. But this definition right here is the standard definition that people now are trying to change so that they can say that they created life. Next question.
All right. For the undergraduate layperson, what is a good comparison or analogy to distinguish chemistry from life?
Well, I guess you could say you could say a carburetor to a car, and the distinction there is far less different than you would have from chemistry to life. But it's chemistry, life, as these living systems are made up of molecules. So a molecule is a piece of it, but a molecule itself does not, does a carburetor try to become a car? I don't think a carburetor is trying to become a car. You can put a carburetor right next to a car that all it needs is a carburetor, and that carburetor is not going to jump on in there. It's not going to do it. Somebody has to pick that carburetor up and put that carburetor into the car. The carburetor itself is indifferent to the car. And so I guess that would be the analogy. But with any analogy, it speaks of similarities, it speaks of dissimilarities. So there's certainly a lot of dissimilarities there in that analogy that I could think of.
All right. So what is the protein folding problem, and how has AI contributed to solving this problem? Is it relevant to the abiogenesis discussion?
Sure, it's relevant, because if you have a protein, the protein has to fold up into a certain shape to be an active catalytic system, say for an enzyme, or it has to have a certain shape just to be a functional protein of any type. And many proteins are way too big to fold in themselves, so they have chaperone proteins which act upon them, which help them to fold. If you just take a string of amino acids, those often will not fold into the shape they need without the chaperone. And how do you go from primary structure, the sequence, to folded structure? How do you predict that? And that is a massive problem. And people have gotten actually very good at that. There's a guy named Baker who's gotten really good at predicting that sort of thing. So there's been enormous computational effort for over 30 years poured into that. That's the Leventhal paradox. How do you go from primary to this folded? And people are getting really quite good at that. Is AI helping? I'm sure AI is helping now. But this has been a computational problem for a long time, and people are getting really quite good at it now. But how they fold without certain chaperone proteins, we really don't know. And how you go from primary structure to knowing what the folded structure is a complex problem, but people are getting really good at it. That's why I say some things are getting closer, while the overall target is getting further away.
All right. So you mentioned that RNA must be stored at negative 120 degrees Fahrenheit. Is this why the COVID vaccine needs to be stored at a low temperature? And could you comment on the chemistry and mechanism of the new vaccines for COVID?
Okay, so yes, this is precisely why, because they're RNA-based vaccines, and you store them cold to prevent the RNA decomposition. And you can have them at 0 degrees. You can have RNA at 0 degrees, but it's decomposing at a certain time scale. Now, if you look at the Moderna has about 3 times more RNA than does the Pfizer vaccine. And until very recently, Pfizer was saying you have to keep theirs at -80, whereas Moderna said you can keep ours at 0. And I don't know, I'm just guessing that they can keep theirs at 0 because they have 3 times more RNA in it. And so they can afford some decomposition occurring. But more recently, Pfizer has just said, no, we think you could store ours at 0 as well. So probably because the transport is fast enough that at 0 degrees storage, the rate, so if you lose 5% of it, you'd be okay. But remember, when we're talking about origin of life timeframes, even if you're 0 and you're 5% over a period of 4 days, that's a lot. That's a real lot. Now, as far as the vaccine, I know there's been a lot of things people have sent me on the internet that is a bunch of nonsense about, we're going to get injected, the government will be able to track us. Look, I know a lot about nanotechnology. You can't track people through this. There's no power source to do the track, and they'll be able to, will be connected to cryptocurrency. That's all nonsense. But the question is, what is the long-term effects of these? We don't really know. We don't know what the long-term effect of any drug is. Any drug. You take any drug, you don't know what the long-term effect is. This is a new class of drugs. The beautiful thing about an RNA-based drug, now you have the South Africa variant, which can escape what this Moderna drug is going to give you. And Moderna has just come out in 3 weeks. You can modify. It's much faster than the traditional way of building a viral vaccine. A vaccine against a virus. And so you can modify and you can come with the boosters really quickly now. That's the beautiful thing about it. And I can't say a whole lot about the safety of this because I don't know, but I will say that I've been vaccinated and I was happy to be vaccinated. And I feel much better now that I'm vaccinated. So you can do with that as you like, do whatever you feel comfortable with. I'm not encouraging you to be vaccinated. I encourage you not to be vaccinated if you don't want to be.
All right.
Okay.
Thank you for that. So just FYI, everyone, we will be asking questions until around 2:20, because we then have to prepare for the next session at 2:30. So the next question is that the media makes a big deal about finding water on Mars or other planets, but you mentioned that some necessary reactions don't happen in water. This seems contradictory. Why do people think water on other planets is important?
No, that doesn't mean that. So I'm not saying that origin of life requires all reactions to be done in water. Requires some reactions not to be done in water. I'm not saying that. I'm saying what we do here on Earth, when we do a lot of these modifications, the only way we can get them to work is to not use water. The only way we can get hypercycles to work is to not use water. The only way we can get a lot of these reactions where you up your enantiomeric excess is to not use water. That does not mean it could not be done in water. I'm just saying we haven't figured that one out yet. So when people say we got that figured out, yeah, you got it figured out when you don't do it, when you do something that's not prebiotically relevant. Finding water is exciting to people. It's not that exciting to me. You need water for life as we know it. That is certainly the case. And I guess that means that they're one step closer to having life. We've got life on this planet is ubiquitous. Everywhere you turn is life. In fact, it's very hard to find a place where there's not something living. on this planet. It's hard. I mean, there's life is everywhere here. And so nearby planets, if they have life or remains of life, I mean, there's a good chance that it came from this planet, because there's a lot of exchange between planets. And but people get excited with certain things. I'm not an astrophysicist, so it doesn't excite me, but I can understand how they get excited. I get really excited when I find, one new little thing in a molecule, and like, wow. And then people are like, you care about that? My wife is like, you care about that? Yeah, I care about that. So I'm okay with people thinking that.
All right. So you've read a lot of the origin of life research. What are the key terms or phrases that you read in the literature that cause you to be suspicious? For example, you said the word spontaneous is a tell.
Yeah, spontaneous. That's a nasty one, and it's used all the time when you have no answer. But then also, there are extrapolations that are just crazy big. I wish I had some of them I could read to you. I mean that origin of life researchers will come out with, and that make you just scratch your head. How did they get that from the work that they did? And so I give you an example here. I was just reading this one this morning. So there was some work done on what was said to be a prebiotically relevant way of making of stringing together nucleosides, adding the phosphorus and making short nucleotides. And they made a 6-mer, 6 units hooked together, working from a paste, which means that they didn't have the age-old problem is, how do you consider such dilute solutions where you have a molecule over here, say a nucleotide here, and it's got to react with a nucleotide over here? When it's so dilute, the chances of it finding each other is much lower than some decomposition pathway where another molecule hits it. Well, this group, quote unquote, solved that problem by putting the whole thing together in a paste, where you're going to find so many nucleosides together that you can add phosphorus to it and get them to hook up to form oligonucleotides, is remarkable. So they solved it by just sticking a whole bunch together with no explanation of where they came together. But here's what the lead author wrote, or was quoted as saying in a magazine, in an article. He says, "This finding is an important step toward the development of a detailed chemical model of how the first life forms originated on Earth." What does that have to do with a detailed model of how first life forms originated on Earth? So you look at some of the claims that are in there, and they'll put, he had similar claims in his own article, and in the actual article. And you look at it and you say, how did what you did affect this? But people will only read those sort of statements, not understanding the chemistry that was done was very, very simple and mundane. And I would argue the chemistry is so simple, but you put so much irrelevant backing into it, prebiotically irrelevant material into it, that's probably how it wasn't done. That's all we can say about it. So it's the extrapolatory claims that are evident in those articles. Those are the ones that you got to be careful of. You have to be able to look at the data and to show, like what Sutherland did. He says all cellular subsystems could have formed from one little piece of chemistry. All cellular subsystems. I mean, that's like from here to the end of the universe extrapolation. Okay.
All right. So this is the last question I think we'll have time for. So sorry everyone else that we didn't get to your questions, but we were under a bit of a time constraint. So the last question is this: is the genetic code a property of chemistry or outside of chemistry? Do we have any idea how the genetic code originated?
No, we don't. Now if you just string together these nucleotides, you will get information, this Shannon-like information that I mean, these 3 letters form 3-letter codes, but that's not relevant to life. It's not relevant to life. That's just background noise. That's not relevant to life. The relevant to life information, we have no idea where it came from. That is not a function of chemistry. Chemistry doesn't move in that direction. Okay?
All right. Well, I think that unfortunately is all the time we have for this session, but Dr. Torre, thank you so much for sharing your knowledge of the chemistry behind this question with us. I really appreciate it. I think that was very, very interesting. And I would just encourage you all, definitely check out Dr. Torre more. He's got those lectures posted on YouTube. And he really was quite serious when he said that if you don't know Jesus and you are interested about learning a little more about Christianity, he certainly was quite serious about meeting with you one-on-one. So definitely send him an email if you're interested in that. But Dr. Torre, thank you so much for your time. We really appreciate it.
Thank you so much.
Absolutely. Very good. Well, I would like to announce the winners from our prize drawing. So the winner from the Mystery of Life's Origin book, which Dr. Torre was a co-author was won by Derry Sang. And the winner of our $20 Nutramax gift card is Thomas Lynch. So we have been in communication with both of them to let them know how we're going to get those prize winnings to them. But our next session at the end, we will do another prize drawing for a book and a gift card as well. So hang on for that. We are going to take a quick break, and we will be back here at 2:30 Eastern time. So see you in a few minutes.